Welcome back to The Explainer. Today, we're diving into a topic that, honestly, most of us try our hardest to ignore. But the truth is, it's absolutely fundamental to human biology. We're going to completely reframe how you think about the end of life by looking at the highly structured, almost architectural chemical process of death. It's an incredible story of transformation, cellular resilience, and some seriously complex chemistry that happens after the heart stops beating. Let's get right into it.
So let's kick things off with a question that challenges a pretty universally held belief. Is death just a single instantaneous moment? I mean, most of us think of death like a light switch, right? The heart stops, the switch flips off, and that's the whole shebang. Everything just goes dark at once. But modern science is actually proving that this common perception is completely inaccurate. Death is not a solitary event. It is an ongoing, sequential process. In fact, researchers at NYU Langone Health uncovered a massive, paradigm-shifting truth. Cells in our brain actually undergo their own distinct process of death. And get this, that process can persist for hours or even days after they're deprived of blood flow and oxygen. There was even a landmark study back in 2001 showing that human brain biopsy samples taken several hours post-mortem could literally still be grown in a laboratory. The cellular building blocks of who we are don't just immediately surrender. They are incredibly stubborn.
To really grasp those crucial first moments after the heart stops, think of it like this. Normally, a localized stroke happens when a blood clot starves just one specific region of the brain of oxygen. We know that if doctors can restore blood flow fast enough, those cells can be saved. Well, from the brain's perspective, cardiac arrest is basically a global stroke. It cuts off oxygen to the entire brain all at once. But just like a localized stroke, if blood flow can be restored in time, those resilient cells across the whole brain might actually still be salvageable. However, we do inevitably hit a biological point of no return. And once that threshold is crossed, the body begins a highly structured chronological journey mapped out by forensic experts like Dr. Prasad. It starts right at 0 minutes when circulation and respiration stop. Then, within the first 24 hours, we see early changes. The body cools down in a process called algor mortis, blood settles due to gravity in post-mortem hypostasis, and muscles stiffen up, which you probably know as rigor mortis. Finally, from days extending into years, we enter the late changes: decomposition, tissue preservation like adipocere formation, or even mummification. So, let's zoom in to the microscopic level. This timeline really kicks off with a phase called autolysis. When your cells are definitively starved of oxygen, their membranes just break down. This causes them to literally burst open, spilling all of their internal enzymes everywhere. Without oxygen to keep things in check, these enzymes start digesting the body's tissues from the inside out. It's a completely sterile, aseptic process. The body essentially starts eating itself. And that internal sterile breakdown paves the exact way for the next major phase: putrefaction. While autolysis happens without any bacteria, putrefaction is all about biology. Those self-digesting cells create the ultimate nutrient-rich environment for anaerobic bacteria, the kind that thrive without oxygen, to just explode in population. These microorganisms spread like wildfire through the blood vessels, using our proteins and carbs as an all-you-can-eat buffet to break complex tissues down into simpler compounds. Now, while autolysis is invisible to us, putrefaction eventually makes this wild chemistry visible to the naked eye. The very first external sign of this bacterial takeover is a greenish discoloration right on the abdomen, specifically in an area called the right iliac fossa. You might be wondering why there? Well, that's where a part of your large intestine teeming with bacteria is closest to the skin. The bacteria produce hydrogen sulfide gas, which mixes with breaking down red blood cells to create a dark green chemical called sulfhemoglobin, and that is what stains the tissues. When we dive into the fate of our body's proteins, which make up about 20% of who we are, their chemical destiny is fascinating. Through a process called enzymatic proteolysis, proteins are broken apart into simpler pieces called peptides and then into amino acids. Depending on the specific chemical pathway, those amino acids are then dismantled even further, shedding carbon or nitrogen molecules until they turn into organic acids, biogenic amines, and eventually gases like methane and carbon dioxide. The sensory result of all that protein breakdown, it is incredibly visceral. Those biogenic amines we just mentioned are totally responsible for the distinct smells we associate with death. For example, when the amino acid lysine breaks down, it creates cadaverine, which smells exactly like putrid, decaying flesh. L-ornithine breaks down into putrescine, creating a highly nauseating odor.
These chemicals are so potent that even in the very first minutes after death, way before humans can smell a thing, flies can detect them using specialized antennas and will immediately zero in on the area.
Moving on to our carbohydrates. Now, they make up a smaller percentage of our body, but they put on a seriously dramatic chemical show. Microorganisms quickly convert stored sugars into glucose, which is then fermented into various organic acids, like lactic and butyric acid, plus a bunch of alcohols. Crucially, though, this bacterial fermentation produces a massive amount of gas: hydrogen, carbon dioxide, and methane. This intense buildup is exactly what causes a corpse to visibly bloat from immense internal pressure. And finally, we have the fate of our lipids, or our fats. By using enzymes called lipases, triglycerides are split into saturated and unsaturated fatty acids. What happens next really depends on where the body is. In an environment with plenty of oxygen, these turn into highly fragrant compounds. But in anaerobic, oxygen-deprived conditions, like being buried deep in wet soil, a fascinating chemical conversion happens. Unsaturated fats undergo a process to form a solid, waxy mass of saturated fatty acids, called adipocere. It's also known as grave wax, and it can literally preserve the shape of the body for years. So to bring all this complex chemistry together, we can look at the overarching narrative of human decay. Your proteins undergo proteolysis to release those putrid amines and ammonia. Your carbohydrates ferment to produce alcohols, acids, and bloating gases. And your lipids break down through hydrolysis into volatile fatty acids and potentially that waxy adipocere. Our incredibly complex biology is systematically disassembled into its most basic chemical blocks. But here is where the story shifts, and things get really unpredictable. This orderly cascade of chemical reactions does not happen at a single uniform speed. The actual rate of decomposition is heavily dictated by a unique collision between the particularities of the body itself and the particularities of its environment. Everything from the clothes someone was wearing to the pH of the soil they are lying on acts as a chemical catalyst or an inhibitor. Let's look at what actually hits the fast-forward button on decay. High environmental temperatures and high humidity, they create a literal incubator for bacterial growth. But biological factors prior to death matter a ton too. If someone died with sepsis, their core temperature is elevated, speeding up all those enzymatic reactions. If they had congestive heart failure, their body has high internal hydration from edema, giving putrefaction the perfect moist environment to spread. Even obesity and wearing warm clothing act as insulation, keeping the body's internal furnace burning just long enough to accelerate bacterial action. On the flip side, what surprising elements actually slam the brakes on this chemistry? Well, freezing temperatures, of course, halt bacterial growth entirely. But check out some of these other factors. Wearing really tight clothing can physically restrict the bloating and tissue expansion that's necessary for active decay. And get this, simply placing a body on a cold, metallic surface conducts heat away from the corpse so rapidly that both autolysis and putrefaction are severely delayed. When you combine all of these complex breakdowns of proteins, carbs, and fats, and then mix them with all those environmental variables, the chemical output is honestly staggering. Researchers using advanced gas chromatography have pinpointed an astonishing 832 distinct volatile organic compounds, or VOCs, released into the environment during just the active decay stage of a single human body. 832? We are, in our passing, a massive emitting chemical factory. Now, why does analyzing these 832 compounds matter to anyone outside of a chemistry lab? Because understanding this exact biological breakdown forms the absolute foundation of an amazing field called human taphonomy. This isn't just morbid curiosity, guys. This is highly rigorous applied science. By cataloging the exact chemical signatures of decay, scientists can literally read a decomposing body like a clock and the surrounding environment like a map. This science of taphonomy is put to work in the real world every day to solve mysteries and save lives. Forensic detectives rely on the specific sequential release of these chemicals to estimate the postmortem interval, which is the time since death. Certain volatile fatty acids only seep into the soil at very specific stages, so they act as a chemical timer. Also, by identifying these exact odor signatures, we can train rescue dogs to find victims trapped deep under earthquake rubble, or help law enforcement spot hidden graves just based on how the vegetation above ground reacts to sudden spikes in nitrogen and ammonia. And the chemical footprint we leave behind is incredibly enduring. The impact we have on the Earth doesn't just end when the soft tissue is gone. Studies have shown that the chemical signatures from human decomposition can remain detectable in the surrounding soil for up to 16 years after death. 16 years!
Long after a body has completely skeletonized, the ghost of its chemical decomposition continues to subtly alter the ecosystem around it.
Which brings us to a pretty profound realization. We started by learning that death is absolutely not a single flipped switch, but a cascade of resilient cellular survival and eventual structured breakdown. It is a process that interacts intensely with the environment, churning out hundreds of chemical compounds that can linger in the earth for over a decade. So it makes you wonder, what incredible story will your chemistry tell the world long after you've crossed that point of no return?
Wow, we're already at the end of the video. Come on, watch other fun videos too, and don't forget to click subscribe to the BrainBubble channel.